Thanks a lot. It's a real pleasure to be here, and thanks for the very nice introduction. We're grateful for the work that we do with this organization. So I want to just let you know how much we appreciate the dialogue that we've had over the years. And I've had a dialogue with the leadership of this organization over many years. I was at FDA during the early days of when this organization got started. And when we would cell and gene therapy and regenerative medicine really were more conceptual than realities. At the time, we were contemplating the first approval of the first cell-based therapy at the time, and so we've come a very long way. I want to just share some thoughts on where I think we're going, what FDA is focused on right now. We're at a key point, in my view, when it comes to cell and gene therapy. These therapies now have the potential to address hundreds, if not thousands, of different rare and common diseases. For a long time, they were largely theoretical constructs. Now they're a therapeutic reality, and it's my expectation that they will soon become the mainstay of how we treat a wide range of illnesses. We've seen a turning point in science that opened the door to these opportunities. One, I think, was the advent of effective vectors to deliver gene cassettes. I compare this to the progression of science when it came to monoclonal antibodies. The ability at that time to humanize and develop fully human antibodies was really an inflection point in that science that ushered in the antibody as a mainstay of drug therapy. And so in that case, a product innovation revolutionized a therapeutic opportunity. And I think the same general idea holds true for gene therapy. The advent of reliable vectors is a turning point in the development of gene therapy and CAR T-cell approaches. But these opportunities also present new challenges, in particular for the FDA. To advance this progress and address these challenges, the FDA is conducting applied scientific research in cell and gene therapy on novel clinical trial designs. We're also working to expedite development programs through our use of all of our regulatory pathways, and this includes our Breakthrough Therapy designation and, more recently, the Regenerative Medicine Advanced Therapy designation, or the RMAT designation. We advanced draft guidance on how we intend to apply the RMAT designation, along with other new steps that we intend to take to expedite the development of novel products as part of our regenerative medicine framework that we released in November of 2017. And we'll be releasing a similar framework very soon that will explain how we intend to address manufacturing issues in the development pathway for gene therapy products. I want to do two things today, if I may. I first want to review the policy we advanced last year for cell-based regenerative medicine, because our efforts there inform what we plan to do when it comes to the second topic I want to address, which is the policy we're going to be planning to release on gene therapy very soon. Our November 2017 framework on regenerative medicine clarifies the existing tissue regulations and describes our compliance and enforcement policy for these products. Our goal is fostering the development and availability of safe and effective regenerative medicine products, and it includes the framework by which we intend to focus our enforcement action where necessary to make sure that unsafe products aren't being unlawfully marketed to patients. That framework provides both a push and a pull towards achieving these goals, a carrot and a stick, if you will. It clarifies the parameters that cause products to fall under FDA's pre-market licensing provisions, and it describes an efficient path to developing those products and seeking FDA approval. Under our approach, it may be appropriate for small developers to pool their data as long as they follow common manufacturing protocols. This will give small institutions a more efficient way to meet the evidentiary requirements of the approval process. It'll give them a path for gaining individual licenses for their individual products. But the framework also describes the enforcement steps that we intend to take when products that are subject to our pre-market requirements are being unlawfully marketed and are putting patients at risk. Recently, the agency went to court to seek permanent injunctions to stop 2 stem cell clinics from marketing stem cell products without the required FDA approval, and which were manufactured with significant deviations from current GMP standards. The field of regenerative medicine continues to expand, and we're committed to adjusting our policies along the way as we need to make sure that we're taking measure of evolving risks and benefits that these products offer. As of the end of April of this year, there had been 62 RMAT designation submissions since the inception of the program in December of 2016, and 19 designations have been granted. Of these 19 products, 14 also have orphan designation. This shows how the program is promoting the development in the rare disease space. This gets me to what we're doing when it comes to gene therapy. Certain gene therapies may qualify for the RMAT designation. Gene therapies that produce durable effects may be part of the larger class of regenerative medicine products. And the pace of progress in gene therapy has been somewhat breathtaking. A publication by MIT that was recently put out predicted that there would be about 40 gene therapy products approved by FDA by the end of 2022, derived from a pipeline of about 930 products as of 2017. And MIT predicted that 45% of these products would be for products targeting different kinds of cancer. Now, I don't know if their estimates are right or wrong, but I do know that directionally these predictions are correct. Just this past year, we saw the first 3 approvals of gene therapies, 2 cell-based gene therapies for blood cancers, and a directly administered gene therapy to address a form of hereditary retinal dystrophy. Sorry, excuse me. Can't believe I went to medical school. Guess it was a while ago. The promise is very much becoming a reality. These recent product approvals represent just the tip of the iceberg. FDA has more than 500 active investigational new drug applications involving gene therapy products, and we received more than 100 such applications last year alone. This shows the intensity of the scientific work going on in this field. These are remarkable developments. These products have the potential to cure some of the most vexing inherited disorders. These advances provide hope to a broad range of people, from parents and children with rare metabolic defects to individuals with more common ailments like diabetes and heart failure. On the regulatory side, to advance progress when it comes to the development of these gene therapies, as I noted earlier, the FDA intends to release a suite of draft guidances articulating its framework for the manufacturing and clinical development of gene therapy products. A lot of this new guidance will be focused on the product-related issues, but these guidance documents will also provide recommendations for clinical development in certain areas. In contrast to traditional drug review, where 80% of the review is focused on the clinical portion of that process and maybe 20% is focused on the product issues, I'd say that the general principle is almost completely inverted when it comes to gene therapy. The initial clinical efficacy is often established early, and sometimes in very small series of patients. The more challenging questions relate to product and manufacturing and quality, or questions like how do you change or enlarge the gene cassette that you load into a vector before the gene insert will change the conformation of the vector in ways that also fundamentally alter the entire product's safety or performance. There's also the question of durability of response, which often can't be carefully and fully answered in any reasonably sized pre-market trial. For some of these products, there's going to be some uncertainty, even at the time of approval. But these products are initially being aimed at pretty devastating diseases, many of which sometimes are fatal and lack available therapy. In these settings, we've traditionally been willing to accept more uncertainty to facilitate timely access to promising therapies. This is the direction that Congress gave FDA by creating vehicles like the accelerated approval pathway, or the breakthrough pathway, or the designation with the RMAT process. When we adopt these approaches to accelerating the development and approval of highly promising therapies that target unmet needs, but have some uncertainty associated with their safety or benefits, we've typically done it under strict approval standards. These approaches require significant post-market follow-up to answer the theoretical questions that might remain about a product's risk or long-term effectiveness. The accelerated approval pathway lends itself to this kind of careful balance. It gives FDA the opportunity to approve very promising products early in the development process based on a surrogate measure that's reasonably likely to predict clinical benefit. At the same time, it also gives us strong authorities to require post-market studies to confirm this benefit. And so among the guidance documents that we'll unveil as part of our comprehensive policy framework are documents that will lay out potential accelerated approval endpoints for certain gene therapy products. The first therapeutic area we'll focus on is hemophilia, where factor production may be sufficient in some cases as a surrogate measure of benefit where a gene therapy product can potentially normalize factor production. In these settings, showing a reduction in bleeding rates can be confirmed post-approval as we continue to study a product's long-term safety and durability. The other guidance documents that we'll release deal with specific manufacturing and clinical issues related to gene therapy products. By providing clarity to developers on manufacturing parameters, safety measures, and the pathway toward clinical development, the FDA hopes to foster even greater innovative development in this field. Once these draft guidance documents are released, we'll look forward to your comments on them. We value very much the benefit of the input of informed stakeholders, such as this alliance. The challenges we see related to gene therapy products are the sort of questions that we can help resolve by working together. Though gene therapies have the potential to address both common and rare diseases, currently they hold out tremendous hope in our country, especially for those with the more than 7,000 different rare diseases. The challenges in developing products for such rare diseases that may affect anywhere from less than 100 people to sometimes tens of thousands of individuals are apparent to all of us at FDA. Clearly, we need to leverage all the innovative approaches for clinical development that we can, including the use of novel patient-focused endpoints and novel trial designs. Moreover, we need to apply all of these tools and make them available as part of the agency's expedited development programs to facilitate timely development of safe and effective products, including using the expanded provisions for obtaining confirmatory evidence for accelerated approval provided through the RMAT designation and the provisions in the recent legislation of the 21st Century Cures Act. The use of registries and real-world evidence are likely to play an increasingly important role in this respect. And so as part of our fiscal year 2019 budget requests, we're asking for new resources to greatly expand our capability for collecting post-market data when it comes to new drugs. Part of the goal is to move toward a system that allows more real-time surveillance of safety questions after new products are approved. Though clinical development of products for small populations is clearly challenging, there's another potentially greater challenge that currently appears to be slowing the advance of gene therapy. That's the challenge is related to the current limitation in manufacturing capabilities for both cellular and gene therapies. More specifically, there are two issues that need to be addressed here. The first is technical and the second is primarily conceptual, and I want to address both of them if I may in turn. First, on the technical issues. The current process for producing gene therapy vectors, both lentiviruses and adeno-associated viruses, is relatively inefficient. This has led to a situation where there is insufficient manufacturing capacity and very high costs. It can cost up to $250,000 or more to make investigational product to treat one patient in a clinical trial. These challenges delay and, in some cases, even prevent the more widespread development of potentially life-saving medical products. What's needed are more efficient and standardized production processes for developing gene therapy vectors. Toward that end, the FDA is undertaking an initiative internally and in collaboration with a variety of different partners to help improve the yield of cell lines used to produce gene therapy vectors. We're also taking steps to investigate the application of advanced manufacturing technologies such as continuous manufacturing. We hope to help provide tools to expedite the development of these processes and help you work with them in the field. Among other specific measures we're taking, we're actively pursuing new investments and expanding the use of continuous manufacturing platforms, which may be especially relevant when it comes to cell and gene therapies. Again, we have a proposal for how we'd expand continuous manufacturing capabilities as part of our budget request for the 2019 year that's right now before Congress. The second issue that needs to be addressed is primarily conceptual, though there are some technical aspects here as well. Many of those working in the fields of cell and gene therapy are using the pharmaceutical manufacturing paradigm, whereby early stage development is supported by drug substance produced through a pilot process. And once early stage development is successfully completed, a commercial process is developed for late stage development and for eventual marketing. This paradigm worked well and saved time and money when only 1 out of every 10 products eventually made it to the market, and when the ultimate market size was many thousands of millions of individuals. But now we're in a place where clinical trials demonstrating safety and effectiveness for cell and gene therapies may only require a few dozen individuals in order to demonstrate that the products are effective, in some cases safe. For example, consider a gene therapy that replaces a deficient protein, transforming how a patient feels, functions, and survives. Here, the need to transition from a pilot process to a commercial process has the potential to significantly delay access or even lead to abandonment of the development, as the manufacturing transition can be very costly and very challenging. We're now encouraging sponsors, particularly those working with small populations, to consider developing scalable manufacturing processes with inherent quality attributes that can potentially support scale-up and licensure. One way that this might be accomplished technically is through the use of manufacturing cassettes that produce sufficient product for a limited number of individuals, say 10 or 20 people, which might be sufficient to supply an initial clinical trial. And if the product then appears promising, further development and ultimately the licensing of the product could just make use of multiple cassettes. Alternatively, a continuous manufacturing technique could be developed which could incorporate sufficient scale-up capacity to support the move from clinical trials to commercial use. We've already seen some product developers working on gene therapy products start to invest in technologies that could be adopted to these kinds of continuous manufacturing approaches. It's clear that these new technologies are going to transform medicine and human health. Gene therapy was largely a theoretical promise a few decades ago. Now we should not only expect these products to cure disease, but we also ought to demand that they reach this objective. This field is moving very quickly, and the FDA is focused on addressing the challenges in manufacturing and clinical development that come with that speed. We're grateful at FDA for all the input and support that we receive from organizations like yours, and working together, I believe we have the potential to address conditions that were once thought untreatable and bring new hope to many patients. Thanks a lot.
We have time for a couple of questions, if there are some questions in the audience. Identify yourself.
Bob Reddy, Chairman of the Alliance with Hitachi Chemical Advanced Therapeutic Solutions. Thank you very much for all of your support. We have been, as an industry, working with FDA for a long time, as you know, and we really, truly appreciate the partnership we've had with the agency. It's been fantastic. The question that I have is really a global question about that relationship. Clearly, FDA is using its enforcement authority to go after unscrupulous providers of therapies, people who are delivering therapies to patients without the proper clinical data to support that. Clearly, FDA is addressing the manufacturing issues that are close to our heart. Clearly, FDA is helping us in every way to advance this as a partnership, to advance the industry. What can we do as an industry to help you? We know that you've got challenges, you have a lot of issues you've got to deal with. We've been good partners, we think, which is evidenced by you being here. At least that's how we're taking it. How can we continue to do that, or what do we need to do differently to help you help us?
I appreciate the question. I think getting back to that 80-20 question that I sort of posed, or the postulate I put forward, I think it feels very relevant internally right now, because the clinical portion of the review is not the complex portion. In many cases, we're seeing these applications come in, and there's clear efficacy. Now, we don't know if there's going to be long-term off-target effects from some of these constructs. We don't know the durability of the response. There's theoretical questions around that. But in many cases, there's no question that if a gene therapy construct is intending to replace factor, you see the factor production, and it seems durable in the course of the clinical trial. The questions are going to be more around the product-related issues. It's not around the clinical portion of the review. The clinical consult is relatively straightforward. And I think that that's going to require the development of scientific principles in which we can sort of base foundational regulatory policy in a way that's more complex than even creating standards that guide the clinical portion of the review. So if you think about what CDER's historically done, they've had workshops to sort of define the clinical parameters for measuring success against different diseases. What are the optimal targets? How do you measure benefit? Here we're going to need to look at questions of how product-related issues change the attributes of the product in ways that could create the potential for risk, and then how we continue to look at these things in the post-market. So I think a lot of that scientific work that's going to form the foundation of regulation is probably pre-competitive and could be done through a construct where we work with partners. I think some of it's also going to be dependent upon having in place good post-market follow-up, doing something akin to what the medical device industry has done, where they have worked through medical societies and other alliances to try to create prospective registries to continue to follow patients so that we can continue to collect data post-approval, because the ability for us to make decisions to approve products, particularly through an accelerated approval construct, is going to be dependent upon our ability to collect that information post-market, and it's going to be invariably in registry type constructs, open label types of constructs, and I think the community can be very helpful in helping stand up those. In the same way, if you look at what the cardiology community did with respect to TAVR and some of the medical devices, where really the creation of those prospective registries that they helped develop, I think gave the FDA the capability to approve some of those products, perhaps in a fashion that they might not have been able to otherwise.
Thank you.
Dr. Gottlieb, you mentioned earlier the incredible momentum in the space, the number of programs that are advancing into the clinic and hopefully to approval soon. That just the number of those programs and the rate of innovation around the science that we're all trying to advance surely puts an incredible strain on the agency from a resourcing perspective. Could you speak just briefly to your vision for how to ensure the agency, I guess, continues to build the resources it needs to work with us and to help advance these therapies?
Well, I mean, we've been fortunate through the user fee programs to receive support over the years. We've put forward a number of initiatives this year in the FY19 budget from the administration, so I could speak about them. We're actually, advocating additional resources for the agency, upwards of around 10% increase in the budget authority of the agency, largest ever increase put forward by any administration for the FDA in its modern history. And I would say some of those initiatives do incorporate things that are going to help us advance this field broadly. I think what we're trying to do with respect to create a foundation for the development of continuous manufacturing platforms is going to be very important to this field. The ability to have a continuous manufacturing where you can use cassettes that are scalable rather than have to switch to different types of fixed manufacturing platforms, I think is going to lend itself in the way I described in my remarks to the kind of development that goes on here. I think the biggest challenge from a resource standpoint right now, and I think we have the resources to address this, is that, and this is sort of a broader point, is that what we are doing in many ways is trying to, retrofit might be the wrong word, but we're trying to back apply regulation to a field that has grown up. And I think that FDA historically did the right thing by taking a light touch to this field as it developed. But now this field is starting to mature, and it's more important now from a public health standpoint that the FDA step in and start to establish bright lines. And that's going to mean applying regulation to people who haven't previously been regulated. And it's going to mean taking enforcement action in situations where people are doing things where they're clearly crossing the line into a regulated space, and promulgating products that are creating risk, intrathecal delivery of stem cells, other things like that. That's going to require us to invest more in our enforcement activity, which is something that we're committed to doing, and you've seen us do in recent weeks and months. So that's something where we're going to have to make sure that we're resourcing that adequately.
Thank you for those comments, Dr. Gottlieb. Anthony Davies from Dark Horse Consulting. I think we all agree that the potency and the efficacy we're seeing from cell and gene therapies is going to result in an increased number of approvals out of earlier stage clinical trials, out of phase 2B, even phase 2. And especially in this field, we see that the manufacturing processes at this stage of development of a drug are very immature. How is the FDA going to offer guidance for the, if you will, the CMC must-haves for approval at phase 2 when you do not yet have commercial grade manufacturing? And if I could ask you to follow on with a commentary of whether indeed phase 1, phase 2, phase 3, as we all know them, is going to remain appropriate for this field of medicine.
Well, these are the very questions that we're grappling with right now, and we're going to try to provide some clarity on the guidance that we release. I mentioned that the thrust of the policy that we're going to put out are going to deal with these kinds of manufacturing questions and what the appropriate CMC standards are at different points in development, and what's the minimum standard for market entry in an accelerated approval type of setting when you have a product that is potentially curative for an intractable inherited disorder, or highly effective for a debilitating disorder. I mean, we're going to have to make these kinds of accommodations. We've done it on the small molecule side, where you've approved products not on fully commercial scale manufacturing, but you're right that there is a minimum standard that needs to be met with respect to the GMP. So that's what's going to be articulated in these forthcoming guidances. And I invite you all to comment to make sure that we've got it right. As far as whether or not the traditional approach to the 3 phases of clinical research is going to be what we go forward with, I think you're already seeing a paradigm change in other areas of unmet medical need where we've applied constructs like breakthrough therapy designation. If you look at what we're doing in oncology on the small molecule side of our house, we're going towards more basket trials. There's different types of clinical trial designs I think could be applicable to this field, and I think it's incumbent upon people to try to be creative and bring forth those ideas. The one thing about CBER, CBER has always been I think there's more hands-on development work at CBER on some of these products because the products demand that kind of collaborative approach. We resource the applications with more intensity. When you look at the sort of FTE allocations per application in CBER, it's always been higher because of the complexity of the applications that go through that side of our house. And so that also offers more opportunity to interact with the review staff with some of these novel types of approaches to clinical trial design. Sure.
One more question. Commissioner Joe Ternowski from GlaxoSmithKline. I was intrigued and pleased to hear about your ideas about productivity and continuous manufacture. Can you tell us who's leading that initiative within the agency in terms of the cell line improvements and the continuous manufacturing? I specifically the senior person inside CBER, I don't know. I interact with the center leadership, but I know Peter Mark is very focused on this. I don't know who he's delegated to, but I'm sure that there's a SME lead.
So clearly the agency is taking a global leadership position in terms of fostering the development of cell and gene therapy. And I was just curious as to your vision for how you might interact with other regulators around the globe to help facilitate, this kind of innovation in a global setting. We have discussions from time to time with other regulators. I think a lot of them have focused less on some of the product issues that I talked about and more on some of the developing some common parameters for what the approval standards are for different orphan diseases. You saw what we did recently with Gaucher's disease, where we developed a guidance in concert with the EMA. That's where a lot of the work's focused right now. Some of the product-specific issues are relatively new, and I think, also are guided by what we're seeing in the marketplace. I mean, we're seeing, in all candor, we're seeing companies engage in manufacturing changes with their products as they move through the development process, and it's not always clear to us whether those manufacturing changes are to try to, move around some of the stacked royalties that exist in this space, or whether they're designed purely to improve the product performance. And so those kinds of questions, I think, compound the complexity of what we're doing. So that's why I think, my message would be that I think a lot of these product-specific issues are going to be the ones that are more challenging to the agency, and more challenging scientifically to you all probably as well, and the ones we're going to be looking for a lot of outside input on.
Are you able to comment when you might expect the draft guidance to be released, whether it's the end of the year, this summer?
No, it'll be before then. I mean, it'll be within the next couple of months. I mean, these are going through final clearance inside the agency, so this is pretty far along. Thank you.
And it'll be draft guidance and presumably with a public comment period, or more an interim final? Or?
If you want direct final guidance, we could do that, but
no.
It'll be draft guidance. I mean, these are challenging questions. We're really looking for the input on this. We really want a lot of input on these guidance documents.
Thank you. Thank you.
To pose a question, Dr. Gottlieb. You talked about the guidance for hemophilia that you're working on. Can you talk about how you're selecting the indications for specific guidance and what might be in the process?
Yeah, I mean, I think we would benefit from input on that. I would encourage people to submit to the docket some, if there's places where they think we should be developing guidance. But I will say that we looked at hemophilia because we saw a compelling opportunity to reevaluate the endpoint that we could consider for accelerated approval because of the potential response rates with respect to some of the gene therapy products. If you see someone who's capable of normalizing their factor production as a result of a gene therapy construct, is it still appropriate to look at bleeding rates if they're developing normal factor and you can demonstrate it's normal factor and it has the same physiological qualities of innate factor production. So the technology presented a unique opportunity there, and I think that there's other areas where we would have similar opportunities. So we do have sort of a small list of places where we plan to put out additional clinical focused guidance. At the time that we release all these guidances, we're going to articulate that and invite in comment on where we should be focusing and whether those are the right places.
Just a question related to the 7,000 rare diseases, and that's devastating and really challenging to try to resolve that. Have you thought about a center of excellence for rare diseases or something to help understand the clinical trials across different divisions at FDA to facilitate that development?
We're going to, when we announce the restructuring, the reform of the Office of New Drugs later this month, early next month, we're going to be announcing substantial reorganization with respect to CEDAR and the creation of many new divisions. And some of those might be focused exclusively on rare disease areas. I don't want to get too far ahead of myself because then I won't get any press coverage when I announce it. But we are aware of the benefits of having a function focused on rare diseases more broadly. But, I think because, and it gets back to this issue of the clinical versus the sort of product issues. I think with rare diseases there is a consistency of product-related issues and also trial design issues that transcends the therapeutic area. And sometimes those are the more complex aspects of the review. The clinical questions can be established, can be sort of answered by very objective criteria that are well established. There's not a lot of uncertainty. The uncertainty and the complexity is on more of the trial design, the product issues, and so having some focus on those issues in a rare disease type of division, if you will, or some kind of construct makes a lot of sense in today's development environment.
Yeah. Dr. Gottlieb, thank you for your inspiring leadership. Ralph Kern from Brainstorm Cell Therapeutics. I just wanted to ask one more question around the new reorganization, the Office of New Drugs. How will that play across CBER and CEDAR, and how will the agency tap on the deep expertise in different parts of the organization to really bring that consistency of new product development?
It shouldn't have a direct impact on CBER, excuse me. This is focused on the Office of New Drugs and CEDAR, as you know. I think one of the goals of the restructuring is to try to elevate the role of medical reviewers to be scientific medical leaders within their fields, which is what they are. But I think that a lot of the time is spent on process-related issues. We've benchmarked some of the time spent just trying to develop what are sometimes redundant memos as part of the review process. And we're looking for ways to make that the process of drug review much more efficient, to free up review staff to try to increase their mandate and their prerogative and their ability to engage in scientific work within their fields. More guidance development, making sure clinical guidances stay updated, having the capacity to provide more consults across the agency, because the CEDAR staff, the CEDAR medical staff, are really the house staff for the agency in many respects, and they provide medical expertise not just across CEDAR but across the entire agency. I mean, even CVM, even CIFSTAN, they'll provide consults. And so having them have the ability to engage in more of that work. And I think when we look at what we're doing, we think we're going to be able to gain a lot of efficiency that's going to elevate them and give the staff more time to engage in that scientific work. And that is going to bleed into CBER, because a lot of the Clinical portion of the review, or some of the clinical portion of the review for products in CBER are done by CDER experts in those fields. CBER doesn't have the same breadth of very discrete clinical expertise in a lot of areas. They have it in some. They have a lot of oncologists on staff, but there's certain areas where they don't have as many, and they're more dependent upon clinical consults from CDER, and they're going to have more capacity to do that. One more, yeah, thanks.
Last question here. Regarding the innovative clinical trial design, we know Europe for many years has something called scientific advice. Even before you have an IND or a clinical trial, you can talk either at the local country level or with EMA on advice for these novel approaches. It's particularly in cell gene therapy where you may have a dramatic effect on the life of the disease. Compromise that first trial to look at that as being sufficient in some of the patients for approval process early on, rather than waiting for the RMAT breakthrough when you need clinical data already. It may be at that time quite late for these innovative products that can have a dramatic effect on the survival of many of these patients.
So what's the question? I mean, I would say, if you're talking about the construct in Europe, we encourage companies to come in for pre-IND meetings, if that's where you were going, and we do them all the time. I've been on the other side of this, as some of you know, having worked with the industry and worked in venture capital, and I think some companies are sometimes, especially small companies, are sometimes reluctant to come into the agency and engage the agency early for fear that the advice they're going to get is going to somehow stymie what they think the pathway that they want to pursue is, and that might happen from time to time. You might hear something that frustrates what your notion was, but I think more often than not, getting the advice early on from the agency is actually very helpful in my anecdotal experience and having been on both sides of this now. So I would encourage people who are trying to do things innovative and disruptive fields to come in early. We certainly entertain those meetings, the pre-IND meetings and early meetings. Thanks a lot.
Yes.